Now, I would like to make it clear from the outset where I'm coming from. I'm somebody who's had an encounter with God Almighty in the form of Jesus Christ. I've had an encounter with God which has changed my life around about the age of 16 or 17. And I accept the Genesis account of creation because Christ himself obviously accepted it, even though evolution existed in his time. I heard last time I was here that evolution was actually an idea formed from the Hindus. It was actually a Hindu religion, so the Greeks also developed it. No doubt about Christ actually knew about it himself. So that's where I'm coming from. And you will probably say that I'm biased. And the answer to that one is, I am biased. I'll be honest about that. But let's have a look at the other side. This is a statement made by Sir Arthur Keith. Now, he was the guy that wrote the preface to the 100th centenary of Darwin's book. That's The Origin of Species. He's a very, very ardent evolutionist. And I've only discovered by pure serendipity about a couple of months ago at Glasgow that Sir Arthur Keith is the prime suspect of the Piltdown Man hoax. Those of you know how there was a skull found that was supposed to be a missing link between the ape and the human. And it wasn't until quite a number of years later they realized that it was actually a hoax. Somebody had very, very cleverly engineered it. And he was regarded as being the chief suspect. So probably a bit of a rogue. However, he made this statement which I wholeheartedly agree with. And I'm sure all people have to accept that evolution is unproved. It is speculation. And every time I speak about evolution, I always want to make it clear that it is not fact, it is speculation. He also says it's unprovable, and what he meant by that is that you cannot go into a laboratory and produce a series of definitive experiments which clearly prove evolution. Now, of course, this is evolution in the sense of macroevolution. That is, we all ascended from primordial slime. It's not micro, where an animal adapts to its environment and you can see changes in that animal. I accept microevolution, but I do not accept macro. However, it's very interesting to notice his bias. He believes it because the only alternative is creation. And not on the basis of science does he reject creation. He rejects it on the basis of philosophy. So I think it's very fair to say that evolutionists are as biased as creationists. It's probably fair to say that most of them, not all, are atheists. Now, this is how I came into the whole field of evolution. This is Richard Dawkins, Professor Dawkins, which some of you will know is a professor at Oxford University of some sort of bizarre subject like education of science in the public domain. And he claimed that the eye was wired the wrong way round. And when I first came across this claim, I had a very, what you might find, interesting reaction. Because that is exactly what I thought. And what I mean by that is that the cells that detect light are buried underneath a retina so that the light has to pass through the retina before it gets to the light-sensitive cells, these cells being called the photoreceptors. And it would seem obvious to make the eye in such a way that these light-sensitive cells are on the surface so that the first thing that light hits are these photoreceptors. Now, I decided that was the case when I was 12 years old because I read it in my first year in science at school, in secondary school. So I knew exactly where he was coming from and what he was talking about. Now I'm doing it the wrong way around. Modern technology is sort of beating me. All right. Not very good at pressing buttons. Right, now I want to explain this in more detail. Here we have a section of an eye. And what you can imagine is I lie down on the table with my head facing towards you. And then somebody takes a guillotine and just slices through my eye. And this is what you would see. So you've got the clear part, the cornea at the top. I've got this wee little gizmo thing which I'll try and use. There's the lens there, okay? And we've got the rest at the bottom. And light passes through the wee aperture, which we call the pupil, and of course the aperture is formed by the iris on the other side, and hits the retina. Looking at the retina in greater detail, you'll see that it's actually quite a thick layer. So this is the, whoops, daisy, we're going crazy. This is the surface of the retina here, and the light has to pass through all this thickness of retina before it gets to the photoreceptors. Now, that drawing is very misleading because it's not a thick layer. The retina is about 0.1 to 0.5 millimeters in thickness, so it's very, very thin. But I show it at high power, and what you're seeing here is that the photoreceptors are at the bottom, down here, and you've got these masses of cells between the photoreceptors and the final layer of cells which sends the signal to the brain. And light, sorry, whoops, daisy. Right, okay, so light has passed through to hit the photoreceptor at the bottom. And these layers of cells are a miniature brain. There's all sorts of signals that pass between the cells, and they do a lot of processing. They talk to each other, they share data, they switch other cells off, they switch some cells on, and it's just like a mini computer. And the sort of things which they decide before the information gets to the brain is things like they can detect an edge passing across the field of vision. So there's quite a lot of processing of the image happens in the retina before you actually get to the brain itself. And his argument basically is that if you're passing light through all these layers of cells, you're going to lose some of the detail of your image before the photoreceptors see the light. And you get such comments like this. Now, I don't know how many of you have been onto the internet or you've read the books commenting about the eye and the fact that it must have been designed the wrong way round. I find that I could describe that people rail against creation in a manner to me that speaks of what is actually behind them, and I'll leave the comment at that. But one of the things that my reactions to when I was reading all the literature was this. You remember Job in the book of Job? He makes this statement, "Therefore I have uttered what I did not understand, things too wonderful for me, which I knew not." And I think it's fair to say that we only know about probably 0.1% of what there is to be known about the eye. We know very, very little. We know a lot, but it's a tiny fraction of what there is to be known. And we've got to bear that in mind when we look at the eye and not jump to conclusions. Now, this chap, Lumsden, he did a rebuttal of Dawkins' book, and one bit which he said was that the nerve fibre layer, or if you like, the retina, which is in front of the photoreceptor cells, is pretty well completely transparent, and therefore has very little effect upon the image before it actually gets to the photoreceptor cells. So there is actually no disadvantage in having a retina between the light and the photoreceptor cell. I did quite a number of years in transmission electron microscopy, and 8 of these years I actually did in ophthalmology. And when you look at the structure of the retina under a transmission electron microscope, it is quite astounding the amount of detail there actually is. And I could see detail up to something like 100,000 magnifications. And it sort of puts life somewhat into perspective, in that you think that you're a tiny dot in the middle of a vast cosmos. But when you start magnifying yourself to an inordinate degree, you're seeing more and more intricate structure, which I think sort of, well. And that statement I've written there is quite often has been my genuine reaction as I've looked at various specimens which I've had to deal with, all of it being in the human eye. Now, I'd like to show you a very low power picture or a transmission electron micrograph of what the retina actually looks like. Now, this, from the top there to about there, is 0.1 millimeters, right? So this is pretty thin. And I'd like to relate it to the diagram so you know what you're looking at. First of all, you can see at the bottom, there's a row of cells in the diagram along here, this row of cells, which is the same as a row of cells here. Now, I appreciate that at this magnification, you would never have known that that was a row of cells, a single cell, or a monocellular layer. And the reason you cannot see it clearly is it's full of melanin granules. These melanin granules are very important because what happens is that light passes through the retina, hits the photoreceptor cells, and then is absorbed by the melanin granules. And the purpose of that is that if the light was actually reflected, you would lose some of the detail which you have. You get glare. And so this makes a very crisp image. Now, you'll know that cats, particularly at night time, you catch their eyes in light, and you get this tremendous reflection from their eyes. And they have a structure behind the retina they call a tapetum, which basically is a mirror, which causes the light to be reflected back through the retina in the opposite way. So in other words, the light comes from the outside through the retina, hits this mirror, and bounces straight back out the way it came in. And it's very, very beautifully done in such a way that the light almost perfectly is reflected the same way that it came in. And it doubles the chances of the photoreceptor cells detecting the light. And that's why you find that cats and such like animals have very good night sight. So we have the bottom layer. Now this layer has got a technical term which I might use. I don't know, can't remember if I do. It's called the retinal pigment epithelium. And one of my jobs when I was in ophthalmology was removing eyes from dead people. Wonderful. But what you might be surprised to hear is that although they had been dead for several days, this layer of cells was still living. And I would remove this layer of cells from them, and we would grow these cells in the laboratory. So it's very uncanny that the person is dead and buried, and yet his cells live on, as it were. So Frankenstein, eat your heart out. Then you have the next part. And this part I'm showing here is what we call the outer segment of the photoreceptor cell. Now, the outer segment is the actual bit that detects light. And you can see it corresponds to these sort of long, thin, slender, needle-like processes in reality, in the actual picture. Now, I should say that this section that you're seeing under the microscope has been stained with several heavy metals like osmium tetroxide. Osmium tetroxide's fame is that if you're wearing contact lenses and you're using it in a fume hood, you'll find that it fixes your contact lenses to your eye. And you'll know about it when you go out at night time because you see these wonderful halos of the street lights. And when you remove your contact lens, you remove part of the cornea with it. So it's not recommended wearing them when you're using osmium. The other one which we use is a salt of uranium. This is a radioactive substance. And, well, it's obvious what happens when you breathe in some uranium crystals. So I've already got the card from the undertaker, but I don't quite know when I'm going. Now, the reason why I'm saying this is that retina in real life is totally transparent. You're seeing something which has been specifically stained, and I don't want you to get the impression that that's what it actually looks like in real life. It doesn't. It's completely clear. So the next layer I'd mention is the other part of the photoreceptor segment, which we call the inner segment, and you can see it, the sort of bulbous bit at the top. That's actually what we call a cone. Now, I don't want to go down this whole line, but you probably know that there are two types of photoreceptor cells in your eye, rods and cones. And rods basically are cells which detect light, but they only sort of give you it in black and white. And they're very good for low light intensity, like nighttime, whereas cones gives you color, but cones only work in reasonably bright light. And that's why if you're sitting in a room reading in the evening and not aware that it's getting darker, somebody could walk in with a dress on that you hadn't seen before, and you would not be able to tell the color because the light is so dim that although you can see the person, you cannot differentiate colour clearly. So that's the inner segments. Then we have the actual cell body of the photoreceptor cells. And the cell body contains the nucleus. And if you like, the nucleus is the library or the brain of the cell. And it's quite a thick layer. You can see there's many, many nuclei in that layer. And then above it, you have another layer, which we call it the bipolar cell layer. Now, that's making it a bit simplistic. There are more cells than simply bipolar cells, but I'm not wanting to go into all the details of that. But that's where a lot of the processing of the information takes place. And then above that, you have your ganglion cells. And these are the cells which actually send a nerve straight through to the brain, through the optic nerve. And the actual nerves themselves, from the cells, lie at the top of the retina. Now, I'd like to take a small bit of this retina, this area here, and show you what it looks like at higher magnification. So what you're seeing here at the top are the nuclei of the photoreceptor cells. And then in the middle part here, you're seeing the inner segments, and then you're seeing the outer segments, which actually detect the light, and then you're seeing the retinal pigment epithelial cells, the RPE cells. And I think at this magnification, you're beginning to see the individual pigment granules in the RPE cells. Then we rack up the magnification a little bit more. See what you can do at transmission electron microscope. And this is as far as I'm going. I could actually rack it up further than that, but I'll keep things simple. So this is a junction between the inner segment and the outer segment of the RPE, of the photoreceptor cell. Now, it's fair to say, oops, I'm doing this the wrong way. It's fair to say that this is basically like a stack of dinner plates, right? Something like 100, 150 dinner plates in a stack. And the pigments which actually detect light are within these discs. And one of the problems we have with the eye is the damage which is done by ultraviolet light. Now, you'll know that ultraviolet light basically causes skin cancer. Well, it also damages the retina. And that's why, if you want to avoid national service or being called up, you look at the sun directly for a couple of minutes, particularly if you use a telescope, and you burn a hole in the back of your eye, and that's you can't see, so obviously you can't shoot a gun. They tell you to clear off. It's not permanent damage, but it's not recommended either. Now, ultraviolet light damages these plates, with the result is that these plates have to be continually replaced with new ones which are undamaged. And the plates are actually formed up in this region here. So the new plate forms here, and when it's added to the stack, it shoves a plate down, and the whole stack gets shoved down one by one by one by one until it eventually lands up in the retinal pigment epithelium. And I've got a sort of, I think this should work. Try again. There we go. So that's them running down to the bottom, and at the bottom, the RPE cell eats the plate. And that's why you always have 150 plates. You can imagine that if you didn't have an RPE cell, you'd land up with something like 500,000 plates by the time you died, and your eyes would be sticking out the back of your head, so it's not ideal. Now, the RPE cell, not only does it digest the plate, but it sends all the nutrients back to the inner segment. The inner segment being this region here, so that the inner segment has all the raw material to make new plates. So there's this continual cycle. And the RPE cell is getting rid of the specific bits which have been destroyed by ultraviolet. And that's how you're having fresh plates made all the time. Okay. Now, before I do that, this is me going on to explain why I think the vertebrate eye is the way that it is, and it's not wired around the wrong way at all. What I'd like to draw your attention to is, down here we have a blood vessel. Now, I'll explain a little bit more about that in a wee minute, but I just want you to be aware that there's a blood vessel immediately below the retinal pigment epithelium. And what you probably don't quite realize is that this blood vessel is enormous, right? And to give you a scale of it, compare this blood vessel to one which is in the retina, okay? You can see that the ones underneath the RPE are much bigger than the blood vessels you get in the retina. So just put a wee bookmark on that one. Now this is an eyeball in which the white of the eyeball, the outside coat, which we call the sclera, has been removed. And I've had it removed to show you that there's an enormous blood supply immediately below the retina. Now the brown tissue which you're looking at here is what we call the choroid. The vessels in blue are the veins, the vortex veins and their tributaries, and the vessels in red are your arteries. I cannot resist saying this one. You'll notice that there's an artery that runs along here. It's what they call the long posterior ciliary artery. I worked for a professor of eye pathology, and part of the research I was doing, I needed to have an eyeball cut right through the macula. This is the part in the retina which gives you all your vision. And he, being the eye pathologist, it was his job to cut the eyeball open exactly in such a way that it went through the center of the macula. And he rarely got it right. He always made a hash of it. I'm not mentioning his name, obviously. I actually do remember one occasion where he was holding the eyeball very, very carefully, and he's about to slice it when it spinged along the table, off the edge straight into the waste paper basket. It was rescued. Now, I realised that he had a problem because I went down to Bristol, and I realised that these two arteries tell you exactly where you're meant to cut. And in a week, I cut 100 eyes right through the macula. So the question is, what's your problem? Now, I want to take this bit here at the bottom of the eyeball and show you at high magnification. And this is what you see. Now, first of all, you've got a single layer of retinal pigment epithelial cells along the top. And immediately below it, you have a layer of capillaries, a very, very dense layer of capillaries. This is not normal. You don't see this in any other tissue, such an incredibly dense network of capillaries. And then the bigger vessels are the feeder vessels. Like, for example, these are the veins draining the capillaries, and these are the arteries feeding them. Now, I should explain, of course, I realize that there probably are some of you who are not in the biological sciences. And just a very simple explanation that the heart pumps blood through arteries to a tissue. The arteries break up into very, very small diameter vessels, which we call capillaries. And it's these capillaries which actually feed the tissue with the oxygen and the nutrients and are what pick up the waste products and the carbon dioxide. And then as the capillaries leave the tissue, they join together to form veins. And then the veins take the blood back to the heart. And so you have this continual cycle. So that's what I mean by capillaries. Now, this is retinal pigment epithelium along the bottom, the RPE. Degenerate photoreceptor cells, because this was an old person and it was at the very edge of his retina. And this is one of the capillaries. Now, they are enormous. And to get you in, this is exactly the same magnification in the retina. And that little bit there is a capillary. Now, capillaries are used for getting red blood cells passing through very, very close to this tissue. And of course, the red blood cells are what contain hemoglobin, and therefore they carry oxygen to the tissue. A red blood cell has a diameter of something like 7 to 8 microns, whereas capillaries can have a diameter of something like 4.5. In other words, they are narrower than the cell. And the cell has to squeeze itself and worm itself through the capillary. And that's why it's one very good reason why you should be taking your cod liver oil. Because cod liver oil contains omega acids. And these fatty acids, when they get into the membrane of the red blood cell, make it much more easy to squeeze itself through the capillaries. And so you feed your brain with more oxygen. And notice how your intelligence levels increase. I said that. I bought salmon at the hospital canteen. And I said to the wee girl at the till, said, "Did you know that fish gives you brains?" And she says, "No, it doesn't. I don't eat fish and I've got brains." She kind of proved the point. Now, the choriocapillaris, you can see individual red blood cells here. That's a red blood cell in section. That's one end on. This one is one with a hole in the middle because they can look a little bit like donuts. They don't taste like donuts, so don't try it. But you can see that the capillary is absolutely huge, and these cells are not worming their way through the capillary. Now, this is where we start using the sort of the kind of the logic I'd like to speak about. You remember that you've got the retina at the top, the photoreceptors in the middle, and then this enormous blood supply at the bottom. So light is passing through the retina to the photoreceptors, and the blood supply is out of the road. It's not in the pathway of light. Now, if we do what Professor Dawkins suggests, and have the photoreceptor cells on the surface, then on top of the photoreceptor cells, you've got to have this enormous blood supply. In other words, the blood is between the light and the photoreceptor. And blood is one of the worst things for light scatter. Ask anybody who's had a hemorrhage above the retina and you go blind. So that's the first fundamental I'd like to say, that if you have a blood supply between the light and your photoreceptor, you're not going to see at all. Now, why is it the way it is? Well, one of the reasons is that light actually produces heat. Now, those of you who were little boys at one time may remember the days when you got a magnifying glass on the one day of the year where the sun was shining, and you magnify it on a piece of paper, and the great joy of seeing the piece of paper burst into flames. If you were a sick little boy, you would do it to a wasp or something like that. But good little boys did it to bits of paper. And it gives you the understanding that light actually contains a lot of energy. And you're actually focusing light on the back of the eye, and thereby you're heating up the retina. Now, if you let the retina heat up enough, you will damage the proteins. A classic example is you put your egg into the frying pan, you turn up the heat, the nice colourless protein goes white, it coagulates. And this is what can happen in the retina. Now, this isn't quite the whole story, because you'll remember I said that light passes through the retina and it hits this retinal pigment epithelium, and the melanin granules absorb the light. By absorbing the light, they release a lot of heat. And it's been suggested that the reason why you have a very good blood supply to the RPE is to get rid of all the heat which these melanin granules are generating by absorbing light. So that's one of the things to think of. The second thing is that this choriocapillaris, the capillaries underneath the retina, actually suck the retina onto the wall of the eye. Now, some of you might know somebody who's had the misfortune of a retinal detachment. And this is where the retina comes away from the wall of the eye into more a central position. It's a devastating condition in that if you do not get it back onto the wall of the eye pretty quickly, certainly I think it's something like a couple of days, the photoreceptor cells will die and you'll have irreversible loss of vision. It's a very, very serious situation. And the way that the capillaries suck the retina onto the wall of the eye is by what we call hydrostatic pressure. In other words, it's sucking water from the retina into the capillary, and it's this suction which keeps the retina on the wall. Now, I wouldn't like you to have the impression that the retina is in any way attached to the wall of the eye. One of the problems we had, if you open an eyeball, I don't know how many of you actually had the great fun of opening a cow's eye in school, but you'll probably remember that the retina came away from the eyeball just like that, very, very easily, because there's nothing physically that anchors it to the wall of the eye. It's just this sucking pressure of the capillary. So this is another reason why we've got this capillary network. The other thing to think of is that the retina itself does absorb ultraviolet light. The cornea absorbs it, the lens absorbs it, and so do the retina. And all three put together reduce the amount of ultraviolet light that actually gets to the photoreceptor cells. And that increases the life of the cells so that they're not being so quickly damaged by ultraviolet light. So that's a good reason why you'd want a retina between the light and the photoreceptor cell. The retina also acts as a collimator lens. Now, if you're sitting there thinking, I haven't the slightest idea what a collimator lens is, shake hands, because last week I didn't either. And don't worry, I'm going to explain. These are the photoreceptor cells. This is obviously a diagram, and we're seeing the discs. The cells are actually running from left to right, and these are the discs of the cells, and that's a space between cells. Now, if you want to maximize your chances of detecting light by these cells, you've got to get the light going along the long axis of the cell. It's not a question that if the light passes through one disc, bingo, you've got a nerve impulse. It actually has to pass through a number of them until eventually it hits the right molecule to produce the reaction. And of course, what's actually passing through is photons. If the light passes at an angle, you can see that it's decreasing the number of discs through which the light is going to pass. So you need a situation where the light is being focused in such a way that it's passing straight along the photoreceptor cells. Now, those of you who are really on the ball will say, well, wait a minute, that transmission electromagnetograph you showed us, these photoreceptor segments weren't straight down. Well, this is what we call an artifact. When we process for transmission electron microscopy, we do so much chemical modification, physical messing about, like dissecting the eye open, that everything gets moved a bit and you don't see it exactly the way it is. But if you actually manage to do a frozen section, you'll see that they actually do fit very nicely vertically onto the RPE. So this is the light coming from the sun from this side, being focused onto the retina, the yellow here being the retina. And just to follow the right rays, what a collimator lens does is that where light rays are converging, it makes the rays go parallel, and therefore go parallel through or right down the long axis of the outer photoreceptor segments. And this maximizes the chances of light being seen by the segments. So that's what we mean by the retina acting as a collimator lens. Now, one of the arguments which is used about the whole business of the eye being wired the wrong way round is that if you look at the retina of the octopus, it's wired the right way round. And I've got a drawing of this. This is from Yamamoto in 1965, so it's an old paper. Never despise the old papers. Some of them are landmark papers, and this is one of them. Now, we have the light coming in at the top, and the first thing it hits are the photoreceptor cells. Okay? Then it passes through the nerve cells before it hits the back of the eye. Now, an octopus eye is a lot simpler than the human eye, or indeed a vertebrate eye, in that the nerve cells at the bottom, oops, just giving you a preview of what's coming next. The nerve cells at the bottom do not do any processing of the image. They simply send the signal straight to the brain and leave the brain to do all the work. That's why octopuses have sore heads when they look at things. Now, the capillaries which actually feed the photoreceptor cells are actually here. You see that? Okay. And you have the equivalent of a retinal pigment epithelium here. So it's immediately beside the capillaries. And it's suggested that the capillaries are doing the same job. One of the same jobs being that they are removing the heat which is being generated by these melanin granules as they're being lit up. They're also there to supply the inner segments of the photoreceptor cells of all the nutrients that they need. And it's a wee bit strange because the cells which eat the photoreceptor segments actually work their way up the sides and then have a wee cap on the top. So it's quite a different setup. And if you read the literature, people say that, look, how is the vertebrate eye any better than this setup? Now, one of the arguments which they use is that the capillaries below the RPE in the human eye are not very good feeders of the photoreceptor segments because they're too far away. Whereas in the case of the octopus, you can see that the capillaries are exactly where you want them. So it seems to be a more efficient system. They then argue that it's not the choriocapillaries which feed the photoreceptor cells, it's actually the retinal vessels. Now that's where they become croppers, because what I didn't point out to you was that the retinal vessels are significantly further away from the segments than the choriocapillaris. They actually occur in the ganglion cell layer and in the nerve fibers between the ganglion cell layer and the middle bipolar cell layer. Now, not only are they geographically much further away, but they've also got to go through a lot of tissue before they get to the inner segment. Whereas if you do it the other way around, you'll notice that there was a lot of space between the photoreceptor cells, and that allows nutrients to get to the inner segments more easily. So, the question is Dawkins right? Is the eye wired the wrong way round? I don't think so. And you've got a few points to chew on as to why it's not wired the wrong way round. But it's actually a perfect example of design. Right, I'd like to move on to one or two other aspects which I find very interesting about the eye. The next one which I'd like to talk about, I think there's just three aspects I'm going to talk about, is the lens. So you've seen this little picture before. This is me lying on the table with my head guillotined. And you're seeing the lens here. And this is a very, very simple drawing of the lens. It's not showing you a great deal, except it's giving a slight hint at the complexity of it. If you looked at a lens, if I got an eyeball, remember your bullseye, you took the lens out and you just thought, it's just a blob of jelly. I mean, what structure is in that? Well, actually, there's a lot of structure in it, and it's very ingenious. The lens is much better than anything that industry has produced as a lens. You can buy a lens in industry for thousands of pounds, and it does not do the job as well as a lens in the vertebrate eye, for a number of reasons. And one of them I'd like to mention is what we call chromatic aberration. Now, what we mean by that is that if blue light hits the lens, it comes into focus at a set distance from the surface of the lens. The trouble with lenses which are made by industry is that if you use red light, it hits at a different focal point. So what it means is that if you have your photographic negative at the blue band, you'll get your blue light perfectly in focus, but your red light will be fuzzy. It will be out of focus. You cannot get both of them put together. And they've gone to inordinate lengths in doing all sorts of various layers on the lens to try and correct that, and have never quite mastered it. And yet the human eye, or the vertebrate eye, does it. All colours, including ultraviolet, focus on the same plane. Quite remarkable. Now, there's another thing about the lens, is a problem they call spherical aberration. And in spherical aberration, light which hits the center of the lens again comes to a focal point, but it's not the same focal point as light which hits the edges of the lens. And the result is that what you see in the center of your lens will be in focus, but what you see around the edges of your lens are out of focus. And you've seen that with a magnifying glass when you're looking at a newspaper. You'll find at the center of it, the newsprint is perfectly in focus, but as you're moving from the center to the edge, it gets further and further out of focus. And the vertebrate eye manages to focus the light, regardless where it falls in the lens, on exactly the same focal point. So it does not have what we call spherical aberration. How does it do it? Well, I think it's fair to say I've not read literature recently, but I don't think we know as yet how it manages to focus different wavelengths of light at the same point. But we do have an idea how it manages to avoid this. This is a very, very small portion of the lens. The bit round the outside is what we call the capsule. The lens actually sits inside a bag. And you've probably heard of people having what they call an extracapsular cataract extraction, which basically what it means is that they remove the lens and they leave the bag behind. And then they put the new lens into the bag so that the lens is exactly where it should be. And it doesn't sort of take a wee trip all over the vitreous or into your anterior chamber and say, hello, I'm a lens, see me? So quite interesting. Now the capsule is part of the reason why the lens keeps its shape. And when your lens is relaxed, it's actually, let's get this right, he's a fat little guy. No, he's not. I always get this the wrong way around. He's actually very, very thin. He thins out because he's being stretched by a whole series of what we call zonules around the edge of the eyeball, which are pulling him. It's sort of like being on the rack and being stretched, and you're getting thinner by the day. And when you want to look at near something very, very close up, there's a muscle round the edge of the eyeball, what we call a sphincter muscle, which contracts smaller, and so the things which connect to the lens loosen up, and he squeezes down to a little fat bob, and that's when you can actually see something very close to you. And it's a capsule largely that's involved in that. But that's not what we're on about now. What we're actually on about are the cells which actually make up the lens. It's a funny organ in that all tissues in the body, when they make a new cell, they can destroy an old cell and just flush it down the sink, so to speak. That is your vascular system. But the lens has no blood supply, and when it makes a new cell, it's stuck with the old cell, which means that as you get older, you get more and more cells in your lens, and your lens gets bigger and bigger and bigger and bigger. And the theory at the moment is that the amount of protein which is in the cells varies from the center to the edge in such a way that it alters what we call the refractive index. And that basically is what causes light to focus on exactly the same focal point, regardless at which point it falls on. The refractive index at the edge of the cell of the lens is different from the center, and it's what corrects the light to get it onto the same point. So I find it a very ingenious system. The next thing I'd like to speak about is the cornea. The cornea is the front of the eye, which is a window, if you like, the clear part in the front of the eye. And I don't know if you're like me, but as a wee kid, I used to wonder and wonder and wonder and wonder, how on earth could you have a piece of skin that was completely transparent? And it wasn't until I was a 24-year-old looking down a transmission electron microscope, I discovered why your cornea is totally transparent. And that's what I'd like to share with you. So I'm taking a wee bit of the cornea here, and I'm showing you, this is a section of cornea. This is actually a light micrograph because the cornea is much thicker than the lens, than the retina. Which is a good job because that means that when you poke your eye, you don't put your finger right through it. Don't try it, just take my word for it. We're talking in the order of probably a couple of millimeters, 2 or 3 millimeters thick, so it's quite a thick layer. I don't want to go down the line of all about how it does it and so on and so forth, although I cannot resist telling you one or two wee tidbits. One wee tidbit I've got to tell you is that there's a layer of cells at the bottom here, which is actually on the inside of the eyeball. And this layer of cells removes water from the cornea, because if too much water gets in the cornea, it starts, oh, thanks a lot, thanks. It starts going translucent, that is, losing its transparency, and then eventually goes opaque. And that's why if you have the misfortune of looking at a dead animal, you can know how long it's been dead by looking at the cornea, because the more opaque the cornea is, the longer the animal has been dead. I don't want you to get the impression that I run around looking at dead animals to see how opaque their corneas are. That's not one of my hobbies. But in old people, this layer at the bottom gets a little bit frayed at the edges. And that's why an old person, some old people, when they wake up in the morning, their eyes are not quite clear and they're not seeing so well. But once they start moving about a bit, the cornea begins to clear again and everything's absolutely fine. Now I want to take a small bit of this. This again is transmission electron microscopy. That little square there, and show you at higher magnification. And what you're seeing here is that the cornea is made up of these layers. We call these layers laminae. You don't need to know that. But if I take a small, very, very small portion, you can see that I've just done a tiny wee bit here. And then we magnify it, and this is when we get up to the marks of something like 100,000. This is of the order of 100,000 magnifications. And what we're seeing are collagen fibers. Now, put it crudely, collagen fibers are basically the steel rods which you have in concrete. They're the things that give the concrete its tensile strength. That is, if you stretch it, it doesn't break. And the same is true in the cornea. And what I'd like you to notice is that these fibers are of exactly the same diameter as each other. That's the first thing to notice. They're about 50 nanometers in diameter. The other thing I'd like you to notice is that they are perfectly parallel to each other. Okay? Now, the fibers are circular in cross-section, and the section's been cut in such a way that you're going right along the length of the fiber. So these circular fibers are exactly parallel to each other. And the other thing to notice is that the spacing between them is also uniform. Now, those of you who've got a critical eye will realize it's not quite uniform at the ends here. And again, this is tissue processing. So it's an artifact that's been introduced. And one of these seminal papers was produced in the 1960s where this guy explained why the cornea was so transparent. And basically what he said was, it's due to the fact that the fibers are exactly parallel to each other, exactly the same diameter, and exactly the same distance apart, and it formed a grid. And then he went into the whole realm of destructive interference and being half the wavelength of light and so on, but I'm not going to sort of bore you with all that kind of stuff. Just take it read that it's because of these features that the cornea is transparent. And this is unique. If you look at any other connective tissue, you will not see this. You will not see fibers running parallel to each other, you'll not see them uniform diameter, and you'll not see them uniformly spaced apart from each other. And when I see stuff like this, I find this amazing. It is just so utterly precise. Now, you look at any man-made material at this magnification, and it's a dog's breakfast. Nothing that man has made comes anywhere near the beautiful, intricate perfection that you see in something like the cornea. And that, to me, makes me say that I am fearfully and wonderfully made. I might be at the bottom end of the spectrum, but it's still quite amazing. It's not, believe me, I'm not a dog's breakfast. May look like that one. Now this is one of my pièces de résistance when I started doing gross anatomy. This is what you call losing your head. This chap took me something like 2 months to actually make, because I had to take a pair of bone forceps and nibble away all the bits and pieces. And I wasn't doing this for fun. This actually demonstrates all the cranial nerves of the head, exactly where they come from and where they are going to. And it's very, very important for medical students. We spend a lot of time teaching medical students about cranial nerves, because when you get a medical checkup, a thorough medical checkup, they have to go through all the cranial nerves and test them to make sure that they are in a working order. So he's actually looking at you. So this is the front of the head. You see the eyes, and I'm going to then look up underneath him. And this is what we see when we're looking at an angle underneath him. For those of you in the know, we've got the eyes at the top here. That's one eye there. The other eye's just up here. Some of you might have heard of the pituitary gland. That's the pituitary gland there. You'll never have seen it like this, and you'll probably never see it like this again, because every time you take the brain out of the skull, you leave the pituitary behind. It's very, very tricky not to do that. Now, I'm not showing you this just to sort of, because I'm an anatomist and I can't help showing you my wonderful specimens. I've got a very good reason why I'm showing you this. What I'd like to bring up to your attention here is there's a nerve running along here, and it's one of the cranial nerves. And this nerve we call the oculomotor nerve. It doesn't matter what it's called. The importance is that this nerve is supplying muscles. And it's the muscles which use your eyeball. And I've just realized I've made a major boo-boo. I should have shown you a video, but it's on my laptop, so you'll not be seeing it tonight. I'm sorry about that. But this is something which you can do when you go home and you look in the mirror. No, you can't. You've got to get somebody else to do it for you. There are 6 muscles that move the eyeball. And you can see from this picture that the muscles are quite small. They're very, very dinky muscles. Okay? Now, what I want you to do when you go home, or even if you want to do it here, is to look at somebody who looks at a line like the finger as it passes across the front of their vision. And what I want you to do is look at their eyes. And what you will find is that the eyes are not moving at the same speed as each other. One moves faster than the other to keep the object. And what that demonstrates is that you have an incredible precision of control of your eye muscles, because both eyes have to be fixed at the same point as the object moves. If they're slightly apart, you get a double image. So you've got a remarkable degree of control of your eye movement. Now you compare this nerve to another one, this one here. This nerve we call the accessory nerve. And you'll see that the two nerves are of similar thickness. But the accessory nerve operates a much bigger muscle. Operates 2 muscles, but the bigger of the two is this kite-shaped muscle on the back, which we call the trapezius. And this muscle does all sorts of things to your shoulder blades. It lifts them up, it depresses them, it retracts them, it fixes them, it rotates them. It's quite a muscle, you know. And that nerve is supplying this huge muscle. And it's the same diameter as the other nerve that's serving these tiny wee twiddly muscles. And the question is, why are the nerves of the same diameter when their operating muscles are completely different size? And the answer is that your trapezius, you don't need very, very fine control of your trapezius. So that one nerve axon supplies about 3,000 muscle fibers in the trapezius. Whereas if you look at your oculomotor nerve, one nerve fiber supplies 13 nerve fibers in an eye muscle. So it's 13 muscle fibers compared to 3,000. And that's why you get such a massive difference in the degree of control. And it's this kind of stuff, when you start adding it all together, and I've only given you a sort of very superficial view of things, but when you start adding it all together, I have to confess I do not have the faith to believe that it evolved. I just don't have the imagination to encapture that idea. I am left saying it has got to be created. Oh.